You are welcome to Abom Academy.
In this video, we are going to talk about the glycolysis. It is a catabolic pathway where glucose is converted to pyruvate. Let's go straight to the point. This is a glucose structure. It's a cis-carbon molecule. And at the first step, glucose is converted to glucose phosphate. And the enzyme that will catalyze that step is hexokinase. Hexokinase. This enzyme has a high affinity for glucose and a low Km value. And the donor of the phosphate group that was given to glucose is ATP. Let's use P to represent phosphate group. The structure of phosphate group is like this. So let's use P to represent the phosphate group. Now, hexokinase is the enzyme that catalyzes the first step. And this enzyme is very, very important. Now, ATP is required in this reaction. ADP. If you can look carefully, from the first 5 steps of this pathway is the energy requiring phase, and the leftover steps are the energy yielding phase. Now we said that this is glucose, and the product that is formed is glucose 6-phosphate. Now this glucose 6-phosphate is isomerized to fructose 6-phosphate, which we are going to represent here as F6P. And the enzyme that will catalyze that step is the phosphohexoisomerase enzyme. And at the first step, a cofactor is needed, which is a magnesium ion. Also, in the second step, a cofactor is needed, which can be a manganese ion. Now, the third step, fructose 6-phosphate is converted to fructose 1,6-bisphosphate. And ATP is required in this reaction. Losing a phosphate group, that means serving as the donor of the phosphate group to fructose 6-phosphate, forming what? Fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. Now the enzyme that will catalyze that step is phosphofructokinase 1. That's how we are going to abbreviate it. Now, fructose 1,6-bisphosphate will be split to glyceride 3-phosphate into glyceride 3-phosphate and dihydroxyacetone phosphate. And this is where the lysis occurs. This is a committed step in glycolysis. And this is the rate regulatory step in glycolysis. And the PFK1 here, the phosphofructokinase 1, is the rate limiting enzyme in this step. There are 2 of them which we are going to see in the other step. Now, aldolase. Aldolase is going to do the splitting of fructose 1,6-bisphosphate to glyceride 3-phosphate and dihydroxyl acetone phosphate. Now, dihydroxyl acetone phosphate can be converted to glyceride 3-phosphate. And the enzyme that will catalyze that step is called triose phosphoisomerase enzyme. That is the enzyme that will convert the hydroxyl acetone phosphate to glyceride 3-phosphate. And another enzyme will come on board. That enzyme is called glyceride 3-phosphate dehydrogenase enzyme. That enzyme will convert glyceride 3-phosphate to 1,3-bisphosphoglycerate. These enzymes add a phosphate, an inorganic phosphate, to this reaction. Also, a vitamin, which is referred to as niacin, is added into this reaction. And what we are going to yield here is the reduced form of the oxidizing agent. This is a reducing equivalent. We're going to yield NADH.